Okay. Good morning, Yani. Good morning, everyone. Before now, in the previous classes, we tried to get the we tried to look at the target genes for breast cancer using some databases like PCIDB and GEO. So after getting the target genes, we now looked for the site of action of Macaranga bacteria using some other databases like I think we use PubMed and um, another one I can't quite remember now. So after getting the target genes for breast cancer using those sites and then the site of action of Macaranga bacteria, we used an application or a database called something Venn to draw a Venn diagram that showed the common genes where the genes for breast cancer target and Macaranga bacteria coincided. Yes, the common genes. And then I think before then we tried to check for the secondary metabolites that are found in Macaranga bacteria, and then we ran a search using Swiss ADME to know about the drug likeness of those genes. And after we've decided that they have a particular level of drug likeness, that was when we now went ahead to look for the targets of those metabolites. Before we now went ahead to look for the common genes, the overlapping genes between the two of them. Then after that, I think we now went um, to find the PPI interaction. Yeah, the PPI interaction of those common genes using some databases like I think Cytoscape. Yeah, we went to Cytoscape to get the PPI interaction. Then we now took it again to one other application that I downloaded. Can't remember the application, but we took it there. I think String App. Yeah, we downloaded Cytoscape app, and then we ran the PPI interaction network there, and then we did some other things. That's what I can remember. Mm.
All right.
Hello, can you still hear me?
Yes, 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 sure. Thank you very much. You have said it. So today we will just continue. I will share the screen. I hope you can see the screen by now. So this is what we are trying to follow. Uh, the flow chart we are trying to follow. So first of all, like you said, we did the number one, number two, we did the number three, and the number four. So for today, I hope you can see what I'm saying. Can you see the screen?
Yeah. Yes, sir, you can see the
Okay.
screen.
All right. Thank you. So we raised the protein-protein interactions that we did. So today we will talk about, God willing, we will talk about the gene ontology analysis and the KEGG pathway analysis. So that's what we will be doing. We will be dealing with today. Let's quickly go to gene ontology analysis. So I was saying that the gene, and in the beginning, in our first meeting, I said that the gene ontology analysis and the KEGG pathway, generally people use the David uh, database. So if you are wondering what's David database, you can just look for it quickly. So is basically that database that will actually help us to conduct this gene ontology and KEGG data, analysis. But for us, we will be using another database.
Sorry, please. Are we recording this session?
Yes, I'm recording it.
Okay.
Yes. Thank you. So the David stands for database for annotation, visualization, and integrated discovery. So if you are interested in visiting the the website, I will just copy and drop it in the chat for you. But we will not be using this database. But the database that we will be using, the results are the same. It's just that from the David, I was not able to get the, how do you call it, the visualization, the pictures. So the database we will be using is quite simple and user friendly. So that's why I use that database, and it's called Shiny. So we will just look for the Shiny here, as we have been doing. We just if you don't know the website, you can just go in Google and type, or your browser that you use, and type directly what you are looking for inside there, and hope you are good to go. So ShinyGO. So this is the website. So this is actually the website. Shiny, the version that is available now is 0.76. Well, that's what how they call it, and it has been released in April. Sorry, you said?
Sorry, Yannick, I'm not talking to you. Sorry.
Oh, okay. So the
let me mute my microphone.
All right, man. So the last version was released in 2022 in April 19, as you can see. I colored it here, and by the moment you get the video, you will have the pointer, and it will be easier for you to see it. So I will also copy this website here and drop it in the chat. So that's the link to this data base, and this is what we will be using for our analysis. We will now go to our hub genes. So from the protein-protein interaction network that we have constructed, our 10 hub genes that we actually find out are the ones here, and those are the genes that we will be using for further analysis. So we have ADRB2 from the smallest to the largest. Well, after the ORC-B, the size of the nodes are the same. It's just that I didn't uh, pull through with changing the sizes, but in the beginning, from this thing, I just showed in the previous our previous discussion. I showed you how you can change the sizes of the different nodes and made them according to their degree. Well, so ADRB2, I will just come here and look for it. So I have it here. Let me just, these are the hub genes. So we have ORC-B also in the hub genes. We have CCN2 in the hub genes. We have what else? I think we have EGFR in the hub genes. ARC1C3 next to. Okay, we have these genes also in our hub genes. We have NEC2 in our hub genes. PLK1T YMS ORCA. Okay, so we have ORCA in our hub genes. We have this in our hub genes. We have PLK1 in our hub genes. That's 1, 3, 3, 3. That's 9. Is remaining 1 hub genes. ESR1. ESR1. Okay, so these 10 genes are our hub genes. So we will now just have to copy the genes and paste them in the database, and we start running the analysis. So you can even see the description here. It's saying just paste a list of genes and click submit. More adjustments below. Most types of genes IDs accepted. Double check the guest species and adjust. So I have copied my hub genes and I will just paste them here. I will just delete the last non-necessary line there, and I will check that I have my 1, 2, 3, 10 genes. Okay, I can now submit. All right, so the different parameters that the database is providing include the pathway arrangement, the chart. If you have you want to do how do you call is it genetical tree? I cannot remember how you call it.
Phylogenetic tree.
All right, so a network, KEGG, genes, and you have the different parameters above here, but we are interested in KEGG and gene ontology, which is the enrichment. But before that, let's work on our the parameters. Yes, the parameters and the different cutoffs that we want to give to our particular analysis. So, okay, well, let me just remind that when we talk about the gene ontology analysis, we want to see the genes that we are putting in for our analysis. What are the different pathways, biological pathways in which they are included? What are the cellular components that they are bringing forth, and what are the molecular functions in which those our genes of interest are implicated in. Those are the most three the three parameters mostly studied for the gene ontology, but there are other parameters that you can find under gene ontology analysis. If you are using, for example, the David database that we talk about, but if you check different literatures, you will notice that most of the time, in fact, not most of the time, almost all the time, those are the three components that they study when they are doing gene ontology: the biological component, the biological process, pardon, and the cellular component and the molecular function of the genes that you are actually putting in. Okay, so the cutoff that the cutoff we want to take, and this choice is also according to the literature, what is mostly used. So for the FDR cutoff, we will use 0.05 as cutoff, and we will let the number of pathways to be shown on our graph or pictures of the analysis. We will let it to 20. So um, the thought is that the most useful pathways, considerable pathways that our genes will be implicated in, might be the first 10 or the first 20. Yeah, so that's just the understanding we can have for why are we choosing number of pathways to show, just limiting it to 20. Now, by the moment we will say 500, well, it might even be difficult. First of all, 500 and the number 500 pathways that will be shown, is it really relevant? Is it really important? And that's number one. Number two, by the moment we will have the figure, we will not, I don't think we will necessarily have I mean, according to the size that we will get, it will be huge for us to manage. So they say pathway size minimum, let's keep it to 2, and let's keep the maximum also there.
Yannick, please, what's FDR?
Okay, I knew someone will ask that question. Okay, well, I cannot remember very well, but it's just the cut is like a p-value.
Me, what I want to say is that all these things that you guys are talking about, it look like magic to me. So maybe because I'm just joining today.
Yes, ma'am. I think it's because you are just joining today. So after watching the previous video, well, okay, I can just go back to this.
No, don't worry, don't let me derail you. I
No, it will
I
not
will bring
delay, Sha.
myself up
I
to
will just
speed.
explain a bit so
Okay?
that you have an idea of where we are now. So you can see on this particular slide, this is the
Yeah.
flow chart we are trying to follow, and you can see some numbers. Number one in the left, okay, on your screen, left top, number one there.
Sorry, please.
Okay,
Somebody is trying to call me. Let me
okay,
decline.
all right.
Yeah.
So you can see that is numbered number one, number two, number three, till number 8. So this is the flow chart we have been following. So we have done from number 1, number 2 till number 4. Today we are hoping to do number 5 and 6, and later we can continue. So
Okay.
yes, so that's what we are trying to do. By the moment you watch the previous videos, you will be able to catch up. We have tried to explain the things there.
Okay, thank you.
All right, ma'am. So now we can submit our work. Okay, we have submitted self before. So this is the results, the enrichment result of the work that we have submitted, and this is the result that we are getting. But we are actually interested in the charts, right? So we will just go directly on the chart, and we will see this is the chart that we are getting here, and this is showing us the top 20 pathways in which our hop genes are involved in. So when we say pathways, we said that we are talking about the biological pathways that we can have. For example, well, mm, sorry, I'm coming. Okay, so on the chart, you can see on the right in the legend, I mean the caption of the chart, you will see n dot of genes. That just means the number of genes, and they are showing us that the different degrees of um, the number of genes that are implicated. Okay, I mean the nodes, the points that you are looking there, we are watching here. Actually, you will see that the size are different according to the number of genes that are implicated in that particular component, or a particular component or pathway, or a biological process. So when you look at uh, the chart, when you look at the chart, for example, from the top to the bottom, the number 2 tyrosine protein kinase, EGF, etc. receptor. So you will see that the node at the end in blue there, after the line, just following the line, is very small. And if we come back to the number of genes, I mean the legends, you will see that probably that's showing the one gene. But when you come to 1, 3, 4, 5, 6, 7, the seventh component mixed. So like I said before, by the moment you get the video, the pointer will be visible for you and it will be easier to follow. Mixed included microtubule binding and cell cycle. You will see that the node there is larger.
Can I ask a question?
Yes, ma'am.
Sorry, let me quickly pick this call. Hello, Itunu. Yeah, yeah. Oh, okay. I'll ask somebody to open the gate for you now. Yeah. Okay.
Yes, ma'am.
That's the question now.
Yes.
Now the map you showed me earlier. So the whole essence of this thing is what do you call it? Is to do docking, molecular docking.
Oh, well, this whole work, there is a lot of other things apart from the docking. So the docking, as you can notice on your screen,
Yes.
at the bottom right, 7 and 8.
Yes.
So the docking is coming up even at the last steps, but we will still, I mean, we will do it. But is including, our work is including the molecular docking, but it's not limited to the molecular docking.
Okay.
Yes.
What is can I ask, according to the sequence, maybe to be clearer to me?
Yes.
Number 1 is target prediction.
Yes. Now, target predictions, yes, but number 1 on this our chart, target prediction from the metabolites. Yeah, so the metabolites are the targets that they are predicting, the different metabolites of our interested plants or formulation. Now,
yes.
Now in number 2, we also look into the different targets that are involved in our ailments. On the charts,
Okay.
is written neuropathic pain, but we are
Yes.
working with breast cancer.
Okay.
So in number 3, the different targets that you have found in 1 and 2, you put them together and found the common target.
Okay.
The essence of that is that because we are trying to see metabolites, let's say drugs from our plant or formulation that can target the
Yes.
elements of interest. So the essence of
Yes.
finding the common genes is that we believe that if we have the target, a common target for the elements being targeted by the metabolites, it means there are hopes that yes, those are working together. I don't know if I'm making sense.
Yes, you are making sense. You're making sense. If I have any question, I will ask.
All right, so that's what we are trying to do. So now from the moment we found out the the common genes there, we will now start running see the protein interaction in number 4. So that will just help us to see the targets, which targets are actually interrelated. So probably they are the ones because for the occurrence of a particular disease, it's not just one genes that is responsible of the disease. Is there is a network of different genes working together. So that's why we want to see which of the genes that we have found out from the common the common genes are actually really working together, something like that. So that's what we will do in number 4, protein interaction. So by in that number 4, we will now find out, we will still fish out some genes that are more related in terms
Okay,
of
sorry. So what number are we now?
number 5.
Okay, so you have done number 4.
Yes, we've done till number 4.
Oh, okay, it's all right.
Yes. So we found number 4, and we are now in the number 5. That's what we are trying to do now.
Is all right? You can continue. You can
All right.
continue. If I have any question, I'll ask again.
All right. So I was on this mix included microtubule binding, and so I was saying that you can see that the node there is bigger. So it's just probably including 4 or 5 genes. And the different colors is also according to the FDR, the value. But now we can choose to arrange our chart a little bit to make it a bit more comprehensive. And what I choose to do is to sort our pathway by the log FDR. So yeah, and then the x-axis too. Same for the color. And the size, like I have just explained it, the size of the nodes are according to the number of genes. And I will keep the font size to 12, the cycle size to 4. So is telling us here that the color, okay, well, by default, the color red is given to the highest, the pathway or component with the highest number of genes actually. So, and the blue for the lowest number of genes included. We can also choose to change the visualization of our graph by using the chart type. So right now we are having the lollipop, and we can choose to have just the dots plot, or we can have it like bar plot. So depending on yourself, what you want you can actually do some changes. So we can now uh, download this plot. So we come to the bottom of our page and we see download plot. Then we just click on download and you download. You can change the width and the height, and you can download the different formats available are PDF, PNG, and SVG. So you can download in the different formats that you want. Okay, so this is our plot. All right, so this is our plot, and this is how it's presenting itself. And okay, how can we actually uh, attempt to explain this plot? So if we take the first one now, this plot, as we said before, is now arranged from um, according to the number of genes included in each component and also the FDR. So the first component we have here is the cell mixed included microtubule binding and cell cycle. So our genes, our hop genes that we have, most of them are actually participating in cell cycle, the biological process in which they are actually involved, cell cycle, and so on and so forth for the other things. That means the different pathways and components that we have on this graph are the ones that our hub genes are actually implicated in. I will just save this if I want to do a publication. So obviously I need to save the chart. Network farm, geoplot, gene ontology plot. So we are good to go. Okay, we will go for 30 seconds and 1 minute. The time this thing is finished, we will just dive in back right away because our time is up for now. Thank you. Come back in 30 seconds or 1 minute. Come back in 30 seconds or 1 minute. Same link. So we will continue where we stopped. Let me share the screen. All right, so what we have just done, like I said before, in our gene ontology analysis, we are considering here, three main components: the biological process, the cellular component, and the molecular function for our gene ontology. But what we have just done, we have just ran our genes, including all the pathways. All the pathways, that's what we have done. Now, when you are working and you want to publish, for example, and according to the literature, you might need to separate your analysis into those different, at least three components: biological process. You want to see the first 20, the different top pathways, biological process, pardon, in which your genes are involved. You want to see separately the components, or let me say molecular functions, that your genes are involving in. And you also want to see separately the components, the cellular components that your genes are bringing forth. So for doing that, is the same way that we have done here. We will just need to change uh, the parameters here. So on the screen, pathway database, which I'm highlighting here, all available gene sets. That's what we actually did before. Now, if we want to stream it to, okay, under this menu that is popping up here, you will see the different pathways that you can choose there. The gene ontology, instead of going with all available gene sets, so you can just choose biological process, cellular component, molecular, and etc. So we want to do them separately. So we will just choose biological process with the other parameters already set. We just say submit. So the enrichment analysis that we bring, that we will have here now, will only be of the, I would say this one. Sorry, I'm coming. Wow. Wow. All right, so I'm not sure about what's happening when I'm choosing streaming it to a particular pathway. I'm not sure, but that is how it is done. So you will just choose the pathway that you are interested in, a particular one, and you will just proceed as we proceeded before, by asking the enrichment, and then you go to charts, and you get your chart like we did before, and you just download the chart. So I will try and find out later, and if there is anything to explain, I will come back and give the explanation to it. Please give me 1 minute. All right, 1 or 2 minutes. So I was saying that I will repeat because I'm trying to record and I didn't start the recording before. So I was saying that the problem was that we didn't choose uh, the appropriate species before. In this on the database, the website, we can see uh, there is a room for us here to choose the species, and because we are working with human, so we need to choose human as a species here even before submitting our work. So we choose the species human, we paste our genes of interest, and we enter submit after manipulating the different parameters, cutoffs, and all of that there. And so here we are choosing for the pathway database. You can see that we choose GO biological process. So by the moment you choose GO biological process and you submit, the result that you will get will only be tailored to biological process, gene ontology, biological process. So this is what we are getting here, enrichment. And here you can also, not you can, but it is advisable that you download the the results, even apart from the chart. You download the results here. You might need it for description later in your work. So you can see here is suggesting you to download the top pathways shown above. So those that are shown here are just the top pathways for now. Or if you are interested in all the significant pathways, you click on all the significant pathways. So if I say I click on top pathways shown above now, for download, is getting downloaded here and is downloaded in a CSV format. CSV format, and we can use it later. Okay, well, let me just for the purpose of this discussion, let me save it. So I will just copy it and come back in our network, and I paste it here, and I will rename it GO biological process. So that's it. Now we can now go on and do what we have done before for the chart. So this is the chart. We said we want to sort it, the pathway by log FDR, x-axis the same, and the color is the same, and we want our chart to be a bar plot or whatever you yourself you want to have. So on this chart, the pathways, the top 20 pathways that we have here, mitotic, cell cycle phase transition, and cell cycle phase, and all of that. So those are the biological process in which our gene is involved in. So that's a bit of it. And okay, me, I generally like to choose the dot plot, because with the dot or the lolly plot, with this plot you can also see the number of genes that are implicated or involved in a particular pathway, I mean biological process. Yeah, so that's it, and we will go on and just download our plot. So let's save in PNG, and I will copy and come back here. I paste for our this thing. I will rename it GO biological process. So we are through with the biological process now, and we will ask our database to bring forth the cellular component. So this is the chart for the cellular component with the same parameters, and we can download the plot for the cellular component. So please be reminded, always remember to actually choose the species humans. So GO cellular, is it? Yeah, cellular component. Cellular component. So we have downloaded the chart for cellular component. The enrichment, as I said, is also important, and we are interested in the top pathways shown above. So yeah, I copy it and paste in my folder. This is GO cellular component. All right, so we are through with cellular component. The last one we are interested in is now the molecular function. So this is the result for the molecular function. We as well download it too. I copy and I paste in my folder. Cellular component, a molecular function. Molecular function. All right, so now we want to see the chart. That's what we will use for our publication, and we download the plot. Copy. Molecular function. GO molecular function. So we are now through with the gene ontology analysis. Please, if there is a question, you can you can ask, and we will try and find out. Yeah, if there is a question. No question. So let's try to go on with the KEGG. All right. So KEGG, KEGG. It stands for Kyoto. Is it Kyoto? Do you say Kyoto or Kaito? I don't know. In Encyclopedia, genes and genomes. Let me check. I wrote it down here. Yeah, Kyoto Encyclopedia of Genes and Genome. So it will also help us to just work with the find out the pathways, different other pathways that are actually our hub genes are actually involved in. I hope. Sorry, are you following me?
Yes, we're
All right.
Yannick,
Okay, so for the KEGG,
sorry, Yannick,
yes.
sorry, what is the main idea of the GO and the KEGG pathway? What's the difference between both of them? Because they are getting the data from the same database. So what's the difference between both of them?
Yeah, all right. So you said the same database, but the KEGG pathways are different from the gene ontology pathways. Okay, let me just open the KEGG differently here from you to see. I hope you can see my screen.
Yes, sir.
Okay.
Yes.
All right. So this is the KEGG. I will just drop this link in the chat.
This KEGG pathway, is it online too? Is it free?
Yes, it's free. I just dropped the link in the chat now. Someone will probably send it in the. Yeah. Joy, Jindima.
Yes, I can hear you, sir.
Okay, so you can read this thing here, right?
Yes, I can.
The one that I highlighted, they say what KEGG pathway is a collection of manually drawn pathway maps representing etc. So the KEGG pathway, you will see, we talk about the metabolic pathways. It will show you genetic information processing and all of that. And generally, what we are interested in doing in our this thing is the pathways, the metabolic pathways, in which the first one, number one here, in which our gene of interest is actually involved in. Do you get? So we are on this how do you call it? On this our website, Shiny website. Despite the fact that both pathways are on that same database, they are different actually databases. I hope you get me.
Yes, I understand. Thank you.
Okay, thank you. Okay, so we are going back to this Shiny. And for the KEGG pathway, in the same drop down menu here, you will see KEGG there. And we just click on KEGG and we bring out the say your gene are highlighted in by downloading pathway diagram is coming up. So this is the pathway. It says select a significant KEGG pathway to show you. So this is the KEGG pathway that we are getting with our genes of interest, and the squares or rectangles in red are actually our own genes that are involved in this pathway. And if you can see the title on this figure saying progesterone mediated oocyte maturation, so it's saying that our genes of interest, according to the KEGG pathway databases, our genes are involved in progesterone mediated oocyte maturation. We can read here that it said select a significant pathway to show diagram with your genes highlighted in red. Now, by the gene ontology analysis that we have just done, okay, for someone who knows about those things, I mean these things, we notice that, at least I noticed that the pathways that was brought up by the gene ontology and even the cellular component, the molecular functions, all those things are mostly involved in a cell cycle. So maybe for my from there I get a clue, and for my KEGG pathway now I can say that I want to see how my genes is actually working in the cell cycle pathway in the KEGG database. So I can just select the cell cycle there, and I get this. You can save this particular figure and yes,
Yannick,
ma.
can I ask you a question?
Yes, ma.
Okay, for example, now the Stachytopheta data that I sent to you yesterday.
Yes, ma.
Like, if I pick the 3 major metabolites from Stachytopheta,
yes, ma.
I want to do molecular docking for all of them. So I will go through this process.
No, Okay, well, the whole thing we are doing now is the network pharmacology. Now, at the end of the network pharmacology, you can include molecular docking to also help you with to see the binding affinity and all those things. Now, you will not, if you want to do molecular docking immediately, directly, you don't necessarily need to go through all these things. So, for example, when you want to do molecular docking, okay, in the example that you give, when I will take the three metabolites that of interest, so it means that probably you know already the target or the proteins against which you want to dock your metabolite. So if you know it, you just go immediately into molecular docking. Please, do you follow, ma? Hello? Anyone following me?
Yes, yes. I think maybe she went offline or something.
Oh,
Maybe our
yeah,
network.
she went off.
Okay,
Yes,
my own question will come maybe. I just want you to, I want to be sure that I don't want to ask questions that you will still answer later. So you can continue, but at least I'm following.
oh, all right, all right. This meeting is 10 to, but we will finish in these 10 minutes. So, so when you actually ask for the KEGG pathway, when you actually ask in the drop down menu, you ask the database to bring forth the pathways according to KEGG database. So in the, you come in the enrichment actually, firstly, and you have your the pathways according to the KEGG database, the different pathways that your genes are actually involved in, and those are the pathways that we have here. 3 and 2, that's 9, 10, 11. Okay, so the pathways involved in which our genes of interest are involved in according to KEGG pathway database include this progesterone, melanocyte, cell cycle, all these pathways involved here. And we can also see that there is breast cancer pathways in cancer, chemical carcinogenesis, and we will actually, like we did for the other ones before, we will download the file. Even my computer. So this is for the KEGG. This is for the KEGG pathway. And then, as we did before, same thing. We will now come and download the plot for the KEGG pathway. I have the download there. I will save it later. And so this is the plot, the chart for this thing, for the KEGG pathway.
Sorry, I want to ask a question.
Yes, please
Very
ask.
quickly.
Yes.
This minus log 10 FDR showing different color, red, purple. What does that mean? I mean, what's the implication of that value?
Okay, is the cutoff is like I said for the FDR is showing a kind of p-value.
Okay, now, so for this color purple, what does it mean? For color red, which is 3.0, color purple 2.0. I
Oh,
mean,
okay.
in understanding, yeah,
Yeah,
what does it mean?
yeah. I said before that this thing you is I don't know. Okay, you cannot necessarily see my pointer here. But okay, now that I try to
I
highlight.
can follow the cursor. Yeah.
So you see that they say color high red and color low this thing. So not only this thing. Okay, but we talk about the number of genes, that's the nodes. But for now, the color is indicating how do you call it? Like, we said that top 20. So it means that according to the cutoff, according to the value, the most, do you say most probable pathway in which our
Okay.
genes are involved in, like stronger evidence. I don't know if I'm making sense.
Okay. Okay.
It's like a
Okay.
ranking. It's like ranking.
Ranking, yes, yes.
Yes.
So the one with red tells us that
Uh,
the
yeah,
genes are more involved.
exactly.
So that means we
The
have higher
probability is
number of our genes. So higher number of our genes are more involved in progesterone-mediated oocyte maturation
okay.
than
Not that higher number of genes. You can see that, for example, you see where they said pathways in
pathways in
cancer,
cancer.
uh,
Also, yeah,
purple there, but the first one here is what, red, but they
red.
seems to be having the same
The same
size of
size.
node. So the size of node is the number of genes involved.
Mm-hmm.
But now the color is just telling us, that they are more involved in quote, they are more involved in progesterone mediated
Mm-hmm.
oocyte maturation than in pathways in cancer. I'm still saying it
Mm-hmm.
in quote.
Yeah.
Yeah, so
Yes.
that's the meaning kind of behind it.
Okay. Thank you. I think I'm
Okay, yeah. But generally, I don't think we have a big deal with that one, as far as um, we get at least kind of, if I can say, what we are looking for, or yeah. So that's it. So this is for the KEGG pathway. I think we will stop here. We will stop here for today. So we were able to do the gene ontology analysis and the KEGG pathway analysis. I will, as usual, upload them later in the Google Drive so that you can have the video and the files. So next time, by God's grace, what we are going to do is now the molecular docking. Now, in the molecular docking, there are several other steps again where you need to prepare the proteins, prepare your ligands and all those things. So probably we will not be able to do it in one day, that same one day, but God will grant us grace and we will see how far we can go. Thank you very much for today.
thank
If
you
there
too.
are questions, we can still be discussing about the questions until my computer went off.
Uh, I think I really appreciate this, and I'm just hoping that we all take advantage of it. Me, I will still need to go through the videos I have previously sent. Because I have been involved in many things, but at least I've been following and I can connect dots on my own. And if I have questions, I
all
would
right.
reach you personally.
All right, sir.
Thank you so much.
Yeah, thank you too. I think bye bye.